Let's take a moment to look at Ctenophora. Look at him. That was a comb jelly, part of the phylum Ctenophora. These little creatures are quite fascinating and can actually be very detrimental to the environment. All that and more on today's biology crash course. Let's begin by defining some of the basic characteristics of this group. The first big aspect of this phylum is the 8 rows of fused cilia that all of them have for propulsion through the water. This propels them like a smooth motorboat gliding across the ocean surface. Just kidding, they're actually pretty slow, and they don't live on the surface anyway, but that's okay for Ctenophora. Other species of Ctenophora flap for propulsion through their marine environment, and others have 2 long tentacles that they can propel themselves through the water. In terms of major groups of these aquatic animals, there are oceanic, coastal, and benthic Ctenophora. Oceanic usually are much weaker and fragile than coastal Ctenophora because the oceanic Ctenophora cannot tolerate the wave motion. They are usually clear or slightly pigmented for camouflage. The coastal Ctenophores engulf their prey and are stronger because they can handle the wave motion. Benthic Ctenophora live much deeper in the ocean and can stand the cold temperatures that are down deep beneath the ocean floor, and they are also bioluminescent. Whoa! Ctenophores reproduce hermaphroditically, meaning that they have both male and female gonads. They actually release sperm and egg into the water at the same time, and the sperm then finds the egg, fertilizes it, then it becomes a whole new Ctenophore right there. They can do this for weeks on end as long as food is readily available, leading to a massive population growth in a very short amount of time. They don't need to be alive very long to start reproducing either, so the population will increase dramatically in the very short amount of time that they're there.
We can suppose that most of the Ctenophora species is self-fertile, which means that it only takes one to make more Ctenophoras.
Ctenophores are carnivorous, and they capture their prey using their tentacles. The principal food of Ctenophores are plankton. However, baroid Ctenophores' main food source is other Ctenophores. Ctenophores have a lifespan that is quite unknown by scientists currently. Some Ctenophores have only lived for 1 month, while others have lived for 3 years, so that is still up in question. The development of Ctenophores is actually much different than most other organisms that we've discovered and looked at so far. There are neither protostomes nor deuterostomes. They're actually diploblasts. Their blastula only consists of 2 different types of tissue, endoderm and ectoderm.
Now moving on to Ctenophora habitat. Ctenophoras, also known as comb jellies, are fairly simple animals that live only in marine waters. They can be found in most marine habitats, from polar to tropical, inshore to offshore, and from near the surface to the very deep ocean. There are about 100 to 150 species of Ctenophoras throughout the world's ocean, although most of these are poorly known. The best known Ctenophoras are those that occur near shore. Such species are typically planktonic, transparent, and non-pigmented.
Ctenophores are eaten by many species of jellyfish as well as by sea turtles and some other fish. Comb jellies have large mouths that engulf its prey. They eat plankton and any type of organisms. They even consume fish and other jellyfish.
We created 2 trees using the software Mesquite. We used character traits as well as 16S rRNA data to create each tree. We were able to find that Ctenophoras are most closely related to Cnidarias in both trees. We also found that Echinodermata and Ctenophora had a common ancestor in the 16S tree, but in the character matrix, Annelid and Ctenophora had a common ancestor. We found that Echinodermata and Chordata were more closely related in the 16S tree, whereas in the character matrix, Echinodermata and Arthropoda were more closely related.
Ctenophora were discovered in the 19th century. It's difficult to say exactly when they were formed due to the lack of the fossil record. However, there is a hypothesis about the group's origin. A very particular paper done by M. Podar and his associates say that the Ctenophores are more closely related to Porifera than Cnidaria, and ancestral Ctenophores resembled modern Ctypids, a globular body with 2 retractable tentacles and 8 comb rows. There's a lot of uncertainty in placement on the tree. We asked why that is, and the most recent classification is based on morphological characteristics. This study uses 18S rRNA to determine. The second study focuses on both 18S rRNA and characteristics in a paper done by K.J. Peterson. We think that the observed tree is different from the journal article tree because we use 16S rRNA instead of 18S rRNA. This has different character traits and species than these two trees in the journal article. The first study used different species of ctenophores, making them more specific than just the general overview. The second study used about 100 characteristics, while we just used 15. So both of the paper trees have much more characteristics and a different RNA strand. Therefore, we can conclude that the two papers are much more in-depth and detailed than ours due to the increased amount of characteristics and different strand of our RNA. We would like to thank the LMU Biology Department, Dr. Ouellette, Dr. Dowd, Tatiana, and Marissa for helping us out with this project. We hope you enjoyed this crash course on ctenophores, and we'll see you next time.